Welcome to Ayan Medical Lecture, and this topic we will discuss about the AIDS. AIDS as the acquired immunodeficiency syndrome is a chronic, potentially life-threatening condition caused by the human immunodeficiency virus. HIV is a sexual transmitted disease. It can be also spread by the contact with infected blood or from mother to children during pregnancy or breastfeeding. There is no cure for HIV, but there is some medication that can dramatically slow the progression of the disease. It starts with the symptom of fever, headache, muscle ache, joint pain, rash, sore throat, swollen limb, gland mainly on the neck, weight loss, ovarious infection. So AIDS is a sexual transmitted disease. It can be transmitted by having sex with infected person, blood transfusion, shaving needle, infected mother to children. The HIV has also been called the human lymphotropic virus type 3. It is a retrovirus, has an RNA genome and a reverse transcriptase enzyme. Using the reverse transcriptase, the virus using its RNA as a template for making complementary DNA, which can integrate into the DNA of the host organism. The CD4 receptor and the GP120, GP41 protein which stick to the CD4 receptor. The HIV virus bend to the CD4 receptor. After some time, it's bend to the CCR5 receptor, which is the chemokine receptor 5. The Robert Gallo at the National Institute of Health believed he was the first to find HIV. But now it's generally accepted that the French physician and esteem discovered the HIV in 1983 to 1984. So anyway, let's talk about the HIV cycle. This is the HIV virus which can bend to the CD4 receptor or sometimes CCR5 receptor. The HIV involves fuses. We have the process of fusion to the cellular membrane and let an HIV virus break up apart, release viral RNA, and perform reverse transcriptase, the newly synthesized DNA. The viral RNA will synthesize the DNA for their own self to make the copies. The DNA is then integrated to the host genome. And then produce a viral protein. Viral RNA that package together and boarding out the cell. The viral protein comes a polyprotein form. This is a multiple protein that can be broken up using a protease, another enzyme. After this process, the mature virus is boarded out, which affect the other cells. This is another enzyme, the protease cleavage. When the scientist was studying about the mammalian immunity, so the scientist given the name, the CD, which means the cluster of differentiation. This one is a mature virion. Here is a question: if a HIV virus damage the CD4s, then why the all immunity is disturbed or destroyed? So CD4s is the helper cells which control and direct the immune response. CD4s recognize foreign antigen and send the chemical signals to the other cells, thereby activating the immune response. So that's why if the CD4 cells are damaged, there will be no chemical signal will send. So that's why the whole immunity is disturbed. Some common complications which is related to the HIV. The most common of the HIV complication is the TB. So the TB is the most common opportunistic infection associated with HIV. The cytomegalovirus, the common herpesvirus, is transmitted in the body fluids such as saliva, blood, urine, semen, and breast milk. The candidiasis, cryptococcal meningitis, the toxoplasmosis. This potentially deadly infection is caused by the Toxoplasma gondii, a parasite spread primarily by cat. Infected cat farts the parasite in their stools. The last one is a cryptosporidosis. Some common cancer which is related to the HIV. Some common cancer which is related to the HIV. The Kaposi sarcoma, a tumor of blood vessel wall. Kaposi sarcoma usually appear as pink, red or purple, lesion on the skin and mouth. And the lymphomas. Other complication. The other complication are the wasting syndrome, neurological complication, and the kidney disease. The last one is a kidney disease. The test which is done for the HIV, the CD4 count which will usually less than the 200. The viral load to measure the amount of virus, and the drug resistance. Some other tests are also done for the confirmation of HIV. The first one is the ELISA test, the enzyme-linked immunosorbent assay, a screening test used to detect infection with HIV. The second one is the Western blot test, a test usually used to confirm the result of an ELISA. And the FR test, indirect fluorescent antibody, a test used like the less expensive Western blot to confirm the result of an ELISA. And the last one which is done for the RNA of HIV, this one is a PCR, polymerase chain reaction. HIV. So the first drug is the Meraviric, inhibit the CCR5 receptor and block the HIV from coming into the cell to begin with. The second one is Zidovudine. This is the first drugs which is discovered for the treatment of HIV. It blocks the action of the reverse transcriptase. The third one is the Veltagravir. It inhibits the integrase process, prevent the viral DNA integration and host genome. The last drugs are lopinavir and ritonavir. which block the protease activity. It prevents the cleavage of viral protein. So, anyway, these were the choice of drugs which can dramatically slow the activity of HIV. The treatment for HIV. So the first one is the non-nucleoside reverse transcriptase inhibitor, which disable a protein needed by HIV to make copies of itself. The second one is the nucleoside reverse transcriptase inhibitor, a faulty version of building block that HIV needs to copies. Some drugs example are here. The third one is the protease inhibitor, which disable a protease, another enzyme. The fourth one is the entry or fusion inhibitor. These drugs block HIV entry into the CD4 cells. The last one is the integrase inhibitor. These drugs work by the disabling integrase, a protein that HIV use to insert as genetic material into the CD4 cells.